Here we have another diagram of translation. Now, this looks a little bit different. And to be honest, there are quite a large variety of different diagrams for DNA replication, transcription, and translation. And they can be kind of tricky unless you memorize what each of the players looks like. Unless you memorize what DNA looks like, what ribosomes look like, what mRNA and tRNA look like. Let's start here at 24. This is the ribosome. Which is made up of RNA, and protein. Now, the ribosome is the location for translation. If you see a long string of letters with U's in it, you're automatically looking at an mRNA. And in order to make a protein, you need a ribosome, you need a gene, right? Because the mRNA carries the information from the original gene that was located on the DNA. But it also needs amino acids. And here we have glycine, phenylalanine, arginine, and so forth. Amino acids. Now, these amino acids are taxied to the ribosome by tRNA. The way I remember it is T for taxi. And the tRNAs have their own, if the tRNAs are taxis, then the mRNA codons represents their different parking spots. So here we have parking spot ACA or codon, mRNA codon ACA. And the parking tag on this tRNA reads UGU. UGU being the anticodon. The anticodon has to match the codon through its corresponding bases. A bonding with U, G with C, and A with U. And together, this would be a polypeptide. Or a protein. A string of 100 to 6,000 amino acids bonded together by peptide bonds. In the last diagram, we were looking at translation. So it was a diagram that was zoomed in out here. This diagram zooms out and shows us both transcription and translation together. Let's start with transcription. Now, how can you recognize this as DNA? Well, one, it's a double helix. And two, its base pair rule is ATGC. DNA having thymine and not uracil. And this is a long string of letters with U's. How can you tell it's an mRNA? Well, it's a single strand, not a double, and it has U's, uracil, instead of T's. Now, this is how it was transcribed. Remember, first helicase would come and unzip the hydrogen bonds of the DNA molecule. Then you have mRNA polymerase putting together the RNA nucleotides onto the DNA template, notice A would have bonded with U. A, U, A, U, G, C, C, G, G, C, A, U, T, A, G, C. Thus copying the gene from the DNA directly onto the mRNA, transcribing it letter for letter, word for word. Then the mRNA is able to leave the nucleus and go out to the ribosome. Now, immediately we must recognize that this molecule here, long string of letters with U's, is the mRNA. Just instead of being pictured inside of the nucleus, we now see it outside of the nucleus at the ribosome. Now, this mRNA is what is used to determine the order of amino acids. Here we have the tRNA carrying the amino acids to the ribosome and lining up its anticodon with the codon. G bonding with C, U with A with U. Then the ribosome puts together all the amino acids to build a protein. Now, back to the analogy we started with, it would be like you have a 600,000 page cookbook in here, the DNA. But you want a single recipe. But the cookbook is 600,000 pages, so it's way too big and heavy to carry out of the library. Frankly, they probably just laid the book down on the foundation and built the library around it. That's how big it is. So, how do you get that information out? You copy it, letter for letter and word for word. You copy down the recipe. So then you can end up walking out with a single recipe and going to your kitchen. At the kitchen, you have all you have your oven, you have all of your culinary tools to make the meal. And you have the recipe, but you can't make a meal without ingredients. So you send your friend, the grocery shopper, out to the store to buy groceries. Your friend brings back the groceries so that you can actually make your meal according to the recipe, producing a gourmet meal. Here's another diagram for transcription translation. And like most transcription translation problems, they start you off with a DNA template strand that is used to transcribe the gene onto the mRNA molecule like this. A bonding with U, C with G, C with G, A with U, A with U, A with U, C with G, C with G, G with C, A with U, G with C, T with A. And then you would translate the mRNA codons to the amino acids by looking at the mRNA codon amino acid chart. UGG, for example, coding for tryptophan. UUU, coding for phenylalanine, and so forth, until all of the amino acids are put in the correct order and come together to form a polypeptide, a protein, a string of amino acids connected by peptide bonds. DNA, RNA, protein trait. In this final diagram representing transcription and translation, we have a picture of the entire cell, highlighting transcription happening here in the nucleus, where the helicase unzipped double helix, forming a template DNA strand right here, and the mRNA being copied in here. Here we have the mRNA molecule leaving the nucleus and snaking its way to the ribosome right here. And here we have the tRNAs bringing these little dots, the amino acids, to the party. The ribosome is reading the mRNA code, and the mRNA is determining which amino acids go in which order. And in the end, the ribosome puts together the amino acids in the correct order until it makes a protein. Transcription and translation. DNA, RNA, protein, trait. Now let's take a look at transcription and translation in a more hands-on sort of way. Here we start with the DNA being unzipped by helicase. So we have a DNA template. The DNA template represents a single gene that's going to be transcribed right through the process of transcription to the mRNA molecules and then translated into a protein. Here we see it again. DNA, RNA, protein, and the protein obviously results in the traits. Now, to help us cut down on any visual mistakes, we're going to draw little lines to divide the code into 3 letters. So we know every 3 letters codes for a single amino acid. Okay, here we go. Almost done. And there. Now, first step is transcription, which happens in the nucleus because the DNA is way too big to leave. A would bond with U because the base pair rule for RNA is AUGC, not ATGC. The only difference is that when you would expect to see a T, in the RNA we see U instead. So notice A with U. T, well, T always bonds with A, and A is found in the RNA. G with C, C with G, G with C, A with U, T with A, G with C, T with A, G with C, A with U, and T with A. And here we have 4 codons. And these are anticodons, which are the 3 letters on the tRNA molecules. And each one of these squares is an amino acid. So, if you were asked to actually find out that the anticodons, it would be pretty easy to do. U would bond with A using the base pair rule AUGC. A with U, C with G, G with C, C with G, U with A with U, C with G, A with U, C with G, U with A with U. But let me ask you a question. Which letters you're going to use to figure out the amino acids up here? The most common mistake that students make is that they use the letters that are closest to the amino acid, the letters on the tRNA. And they would be wrong, because this is an mRNA codon amino acid chart, not tRNA. So we need to use these letters. Always remember, it is the mRNA that is used to code for the amino acids, not the tRNA. Please keep that in mind. That's a silly mistake that costs people a lot of points. Now, here we have codon number 1, UAC. Let's look it up. U, second letter A, third letter C. We have ourselves triosine. T-Y-R. Then we have GCU. GCU right there. And that's alanine. Then we have ACA. ACA codes for threonine. Then we have CUA. CUA, which codes for leucine. And there you have it. The ribosome would then bond all of these amino acids together with peptide bonds. Here's a common transcription translation problem. In these problems, you normally start with a DNA template sequence. TAC, GAG, TGT, TCA, ATC. Now, in order to actually figure out the amino acid sequence, you have to undergo transcription and then translation. Let's try transcription. T would bond with A with U, C with G, G with C, A with U, G with C, T with A, G with C, T with A, T with A, C with G, A with U, A with U, T with A, and C with G. And let's break these up into the mRNA codons. There we go. Every 3 letters. Now translation from mRNA to amino acids. mRNA to protein. AUG, we find AUG over here. Notice, here's AUG. AUG is the start codon. That is the 3 letters that starts every single protein. And it also codes for the amino acid methionine. So M-E-T. The next amino acid is coded for by CUC. So let's find CUC. Here we go. CUC is leucine. So we'll write down leucine. Then ACA. Let's find ACA. Right here, ACA 309. THR. There we go. Then AGU. AGU is over here. And that codes for serine. So SER. Then UAG. UAG is up here, and that is the stop codon. That, along with a UAA and UAG, tell the ribosome to stop making the protein. So stop. Now the ribosome would then bond together these amino acids in the correct order with peptide bonds, forming a polypeptide or a protein.